It's been just an amazing time to meet many of you guys here. Really excited, and thank you so much for the invitation, Mandy and Josh, for inviting me out to give a talk on our research. So I'll begin in this talk. What I'm going to talk about is describe, the type of research that we're doing at Nationwide Children's Hospital. So I run a translational neuroscience lab at Nationwide Children's Hospital. And so what I'm going to describe are some research projects that we've are in the process of doing and have published. And so I haven't, in terms of POGZ research, we haven't delved into that, but we are actively pursuing this area of research. But really what I want to get across is the fact that what we've done previously can be applied to POGZ research as well. So it's really an exciting time to be a neuroscientist. There's so many tools available to us as scientists to help us really understand what's going wrong in disease, especially in neurodevelopmental disorders. So my lab uses stem cells from patients. We can derive stem cells, and you heard this a little bit yesterday in the Simon Searchlight presentation, that the stem cells that my laboratory receives, we can use those stem cells to generate very powerful model systems of the human brain. And we call these organoids, organ-like structures, and specifically brain organoids. So we have a brain organoid or a neural organoid that's shown there. And I'll describe a little bit more about that. I want to give a little background before I move into some of the research. But I just want to emphasize, what kind of tools do we have available to us as researchers? We've heard about mouse models in some talks, but I just want to emphasize, mouse models are enormously beneficial for researchers like myself. But there are key differences between a mouse and a human, not based on the sheer size of the brain. The mouse only has about 71 million neurons, and we have almost about 86 billion neurons. And so even the developmental processes are much different between a mouse and a human. And so we really need to devise human-based model systems to help us understand a little bit more about what's going on in disease. And so this was what my laboratory is doing. And so what I want to kind of talk a little bit more about detail in terms of research. And so for investigators like myself, what tools do we have available? Well, back in the day when I started out as a researcher, back in the late 90s, early 2000s, we really didn't have a lot of tools available to us to study human disease. And that partly because there was no stem cells available to us. So the iPSCs you've heard about, this is a relatively newer innovation back in 2007, that's when it was described. And so, but when I started research, we didn't have these tools available to us. So what we can do is we have mouse models, right? We have some brain tissue from postmortem brain tissue, but there are limitations, right? And so I give a little background of stem cells. So like I mentioned, 2007 was the discovery of these iPSC cells. And so we can take a normal cell, a fibroblast, a skin cell, a blood cell, and push back the clock, so to speak, and push back the clock in terms of their development.
You can keep going. I'll just.
And so we can push back the clock on these cells and make them more stem cell-like, and almost to the fact that they're indistinguishable from stem cells that give rise to all the cell types of the human body. So we call these iPSCs. That's why they're so powerful, because they can turn into neurons, different brain cell types. Very, very important to understand mechanism. That's what we really need to understand, the scientist mechanism of how that mutation is causing a deficit and then eliciting some of these symptoms that we see in patients. But these iPSCs that were discovered back in 2007, another leap of technology around 2013 was discovered, and these are organoids. So these are three-dimensional structures. We grow them in a laboratory petri dish, and I'll show you some pictures of those. And these brain organoids I'll talk to you about are three-dimensional tissue culture models of the human brain. We generate them from iPSCs. So we can't generate them without iPSCs. That's why they're so important to us. And so here we have some brain organoids culture in my laboratory in a multi-well dish there. You can see them without a microscope. They grow in culture. And so what they do, they resemble a lot of the morphological features of the developing brain, which is amazing. So this is a cross-section. We take one of these organoids and we just section it, and we look what's inside. We see very, very exciting morphological features of the developing brain. So inside of all our brains, there are empty areas called ventricles. That's where the CSF is, the cerebrospinal fluid. And lining the ventricles, you can see here in this brain organoid picture, this is called the VZ of the ventricular zone. We can label different cell types in these brain organoids and start to understand what's going wrong. So we, from any individual with the disease, we can start comparing these patient-derived organoids with control organoids that grow healthy and compare and contrast what's going wrong. So a very powerful tool for us as neuroscientists. So let me jump into a project that we published. This was published a few years ago, and I brought this up because it really emphasizes the point of what we can do for POGZ. I am in the Institute for Genomic Medicine at Nationwide Children's Hospital. This project was spearheaded by a very talented trainee in my laboratory, Summer Fair, who's now at USCSD and a medical sciences training program. But in this particular project, a Nationwide Children's Hospital patient was seen by our group, and the patient was diagnosed with a syndrome called OTS2 syndrome. And interestingly enough, OTS2 syndrome patients share very similar symptoms with POGZ patients as well. And so let me just go through that in a few slides. So the patient presented with the following features: profound intellectual disability, microcephaly, epilepsy, and cerebellar hypoplasia, as you can see there in the MRI. So the patient went on a diagnostic odyssey, and the patient underwent whole genome sequencing on the patient, but also healthy parents. And there was a missense change in their DNA, and that went to the OTS2 gene. A one base pair change, which rendered a different amino acid change from a threonine to a proline. Now, we had heard that there's multiple informatics tools. So you have these variants. We have algorithms to help predict, okay, is that pathogenic or likely pathogenic or benign? But again, these are tools. These are not doing research in the laboratory. These are just predictions, these bioinformatic tools. We really need to do research to understand whether that variant is actually causal to the disease or not. And so this is what my laboratory had done. We, from the patient, we generated peripheral blood mononuclear cells, or the cells that we use to derive the stem cells. And we take those PBMCs, we reprogram them, so to speak, what's called the reprogramming process, push back the clock, make them like stem cells. And this is called, these factors are called the Yamanaka factors after the person who discovered this process. And within a matter of 3 weeks, we can generate iPSCs. So very fast process, very efficient. When I started working on this process, it took a long time to do, but now it's pretty straightforward. So we modeled the patient's conditions using brain organoids. And so, so I won't go down kind of deep level in terms of what OTS2 does, but it shares similar functions for POGZ, and the fact that it works on to transactivate genes, whereas POGZ is thought to act on the opposite spectrum as a repressor. But it functions in the nucleus as to regulate genes, downstream genes, and a network of other neurodevelopmental genes as well. And so here's the OTS2 gene, and this is the arrow pointing to that missense change right in the middle of the gene. And so patients I had mentioned show spectrum of intellectual disability, autism, microcephaly, and very similar to POGZ syndrome. Very few patients have been described, 75 patients described. And microcephaly is one of the most consistent clinical features of OX2 syndrome. So how do we do this? How do we go from stem cells all the way to brain organoids? We grow these stem cells in a petri dish. We push them towards the brain organoid lineage with different factors and different medias. And then over time, they grow. And so by 1 month in culture, you can see here in panel F, these organoids we can visualize. We can monitor the growth of these organoids over time. And what we saw was very exciting to us in the very beginning is because these patient organoids were smaller than the control organoids. And we were excited because this is mirroring like this microcephalic symptom in the patients. And so we can quantify the organoids over time, as shown in panel H. And as early as 24 days, we saw a significant growth deficit in these organoids. So this was very exciting to us because now we can model the disease using brain organoids. And so another tool that we have available in the laboratory is called CRISPR genome editing. And you probably have heard about this technique in various news outlets. For this particular approach, we're using CRISPR genome editing to go back and change that single base pair change back to its correct base pair. And so for scientists, this allows us to generate what's called an isogenic control, meaning that making that one base pair change, we now have a very rigorous control, a gold standard control to compare this gene-corrected control with the patient mutation. And so we've done this, and we can do this for any variant. If it's not a significant deletion, if it's a missense change or a frame shift mutation or termination mutation, we can do this quite readily. So what about the data? So we just can look at the organoids and grow, but what else can we do? So we have a lot of techniques in the laboratory. We can analyze how these organoids grow over time. On the single cell level, we can monitor how the cells grow and divide in the organoid. So human brains have what's called, they do have stem cells, progenitor cells that grow and divide. That's how the brain grows, and they differentiate into all various cell types of the human brain, neurons or glia, astrocytes, different types of neurons that say, hey, give a signal or dampen a signal. So this is what we did. We looked inside of these organoids, and we saw, in fact, that the progenitor cells were growing at a much slower rate than our healthy control or isogenic organoids, as shown there. And furthermore, we have another tool available to us. I had mentioned that it's a very cool time to be a neuroscientist because we have these other emerging technologies available to us to help us then study organoids. And so one of these technologies is called single-cell RNA sequencing. So these organoids not just have one cell type or two cell types, they have a dozen different cell types. So all of our human brains consist of like dozens of various cell types working together in a network. So these organoids also have dozens cell types. And so this technology will allow us to look at the single cell level, the gene expression patterns that happen. And so this is very important to us because it helps to pinpoint a defined mechanism of what cell type has gone wrong in these organoids. And so in developing drugs that we can pinpoint to a particular cell type, we would have the ability to do that if we knew the information. And so this technology allows us to take all these organoids, make them to single cells, and capture their unique gene expression profiles. And so what we did for OX2 syndrome, we had done this, and lo and behold, we identified some cells that were absent in these organoids, and these were progenitor cell types that were just absent. And so without this technology, we have no way of knowing this. And so I think this is why it's important that organoids are very cool, but it's the technologies we couple with organoids that help us to get a more informed decision of what's going on. And I'm excited because these very same technologies are being applied to POXI. And not just one variant, but multiple variants, and understanding how those variants impact disease and help us to understand a better picture of why some patients show a spectrum of symptoms. So when we get that data, what does it give us? Well, the human brain is amazingly complex. We're all very complex. And so these gene expression patterns in these cells gives us some idea really what the pathway is. We say molecular pathway, and we were hearing some talks about different drugs working on different receptors to dampen activity. And so if we have this information of pathways, we can start to understand what pathways we can alter to alleviate symptoms in patients. And so what we did with OX2 syndrome, all of this research allowed us to hone in on a particular signaling pathway called the Wnt beta-catenin signaling pathway. And there's a lot known about Wnt beta-catenin pathway. And so it allows us to think about what ways we can modulate the pathway to impact the disease. So now I want to move into POC. And so I mentioned that there are all these tools available to us as neuroscientists. Now you've seen the schematic before. I made the schematic within this past week. But what I want to, all these mutations that are shown in the POC genes are all the specimens that the Simon Searchlight has. And actually, this is fairly old. This is a little bit outdated. This was about 2 months ago. But now we have a lot more patients readily available to us. But do you notice Simon Searchlight has, and I know a lot of you have donated some of the blood for this cause. But I'd like to emphasize that there are a variety of variants that fall in the beginning of the protein, the back, and these well-defined domains. And so one of the open questions in the field is, we identified this variant in my child, and they display these symptoms. Why is it that they display the symptoms and another patient's variant shows a different symptoms? And so what we can do, we can model those variants in these brain organoids and start to understand why is that. And so one way we can think about it is in terms of functionality. You think of 100% functional POC protein. That would be with no mutations. Instead, with a mutation and a frame shift early on, you might expect it to be more severe than one that's later, but we just don't know really. We're just hypothesizing. We really need to do the research. And so I'm really excited, based on expertise my laboratory has established, is to move forward with modeling these variants with brain organoids. And so I had mentioned these techniques we have in the laboratory, and these are just to reiterate again, how we can apply them to POC. And so we have ways to look for different cell types, how they're developing brain organoids, and we have a technique called immunofluorescence. We can label proteins or different biomarkers in the organoids to start understanding mechanism, investigating what's really going on. And we can look at progenitors or excitatory neurons or inhibitory neurons or astrocytes and start to understand, okay, what's wrong in the variant versus the control. And so we can also do single cell RNA sequencing. I talked about that. And then also CRISPR genome editing. And so the CRISPR genome editing is really important because the fact that when we make that gene correction, we run an experiment again, and we would expect those would be exactly very similar to our controls, our healthy normal controls. And so it gives us that cause-effect linkage and reassurance that mutation is causing those variants in our brain organ model. And so I'll just wrap up and talk about, truly this is a team science approach. I'm very fortunate to have a very strong team and strong collaborators. Craig McElroy and Allison Bradbury, key collaborators. But it's been an amazing environment to do research. And so I think it's very important to collaborate and to share amongst the community. And I'm so excited to have met you and be connected with all of you as well. Happy to answer any questions you might have.
So I will share, we're looking at raising funds to hire Dr. Hester and his team to help further POGS research. So with that in mind, we'll be talking about raising funds tomorrow and how we move forward with that, because it's a heavy lift and we can't just do it for 1 year. We've got to continually be able to do it. So on that note, we do have questions.
With the CRISPR gene editing, would that, for someone that's already born, like our children, would that benefit them at all if they did get the gene editing done? Would they still have the same, I would say their physical appearance would still be the same and everything, so it's not going to completely fix, certain things, but would that help them in any way, do you think? Or how does that work? Does it have to be caught before birth, or?
Very good question. One way to think about that, whether POGZ is really important during brain development, but there's a lot of hope because in some other studies, and actually in POGZ, one of the mouse models, I've seen that some of these phenotypes are reversible. And so it makes me hopeful that correcting that gene, so it's also important in all of us adults after brain development. It controls downstream genes important for how neurons communicate with each other. So I'm very hopeful that restoring it after brain development, significant milestones in brain development, that there will be some reversible symptoms. The CRISPR genome editing technology is a very exciting one. And thinking about, we've had some success stories with metabolic disorders using CRISPR. In the brain, it's very challenging to get that machinery back in the brain. So there are a lot of challenges with that approach. But thinking about those technologies and medical interventions that get right to the root cause of the condition, CRISPR genome editing is the one, because you really go to the genetic mutation. And so I'm very excited to see that field to move forward. But that's a great question. That's why we need to do the research, develop the mouse models, develop the human brain models, human brain organoid models. One thing I will say that with human brain organoids, we can also answer that question. We can deliver back a normal copy of the POGZ gene to the brain organoids after they show a phenotype abnormality, and then ask, okay, can we reverse those in human brain organoids? And so we can do that as well. And so there's a lot of things we can address with the brain organoids. Yeah.
Very interesting. Thank you for being here, coming aboard alongside us.
Thank you so very much.